So today we will continue our second lecture, and first we will start with disorders of imprinting. So we already talked about imprinting when we were talking about uniparental disomy. Today we will dive a little bit deeper into imprinting physiology. So here on this picture you can see epigenetic reprogramming in the germline cells, right? So what's happening here? When you receive one copy of your chromosome from your father, the other one from your mother, in like the stage of primordial germ cells, you actually have really similar features in both parts of the chromosome, right? So when they actually differentiate and become gametes, process of imprinting kicks in, and what that does, it inactivates one of these genes, right? So in the case of paternal chromosome here, you have gene number 1 active and gene number 2 inactive. In the case of maternal chromosome, you have gene number 1 inactive and gene number 2 active, right? So in order for cell to fully function, it have to receive gene number 1 and gene number 2 in an active form. So therefore, you have to receive one copy of your chromosome from your father and the other one from your mother. So when it becomes pathology, we already mentioned Prader-Willi syndrome and Angelman syndrome. Here we will discuss about imprinting more. So for example, when you delete part of your gene from your paternal chromosome, you receive only gene number 2 products because gene number 1 is inactive in maternal chromosome, right? So you develop Prader-Willi syndrome. In the case of uniparental disomy, when you receive both of your chromosomes from your mother, you again getting only second gene products because first gene is inactivated, and again you develop Prader-Willi syndrome. So in the case of Angelman syndrome, when you delete your maternal genes from maternal chromosome, you receive only first gene products because second gene is inactive, imprinted, right? And in the case of uniparental disomy from paternal side, right? Again, you get only first gene products because second gene is imprinted and inactive. So this is when you develop Angelman syndrome. Maternal chromosomes losing, it's Angelman syndrome. Paternal chromosomes or genes are losing, it is Prader-Willi syndrome. That's it. So in the case of Angelman syndrome, we know the name of the gene. You have to know that as well. It's UBE3A, right? So now let's talk about presentation of each disease, right? In the case of Prader-Willi syndrome, first what comes in mind is obesity. Why are these children or adults are obese? Because they eat a lot. So overeating, right? Second one. So how you can see Prader-Willi in newborn, for example, you will see presentation of hypogonadism, and you will see flappy baby syndrome when hypotonia is present, right? So in these children, you will see hypotonia, hyper eating, right? Then hyperweight, obesity, right? And hypogonadism. You can memorize like this if it's comfortable for you. So 4 H's, right? For Angelman syndrome, first what comes in mind is inappropriate laughter, because these children are so kind and just naive, let's say, right? So in these children, we have mnemonic for memorize the other features called hails, like this child hails the angels, for example, hails the angels, right? Where H stands for hand flapping, A stands for ataxia, E stands for intellectual disability. Both of these children have intellectual disability. L stands for laughter, inappropriate laughter, and S stands for seizures. That's it for these two diseases. Now let's look at the example here. A 4-year-old girl has short stature. Both of her parents are of average stature. Genetic analysis shows paternal uniparental disomy of a particular locus. Which of the following mechanism might explain the short stature? So we have paternal uniparental disomy here, right? So maternal, we exclude immediately. Then we look at the answer choices C and D. It says paternal imprinting of one allele or paternal imprinting of two alleles. So as mentioned in the stem here, we receive two of our chromosomes from our fathers, right? So we look at the case here. Two of these genes, of genes number 1, are imprinted, and therefore we get only second gene product, right? But in order to have average stature, we need to have two genes products, and therefore this child developed short stature in comparison to his or her parents, right? So therefore we look for two alleles being imprinted, and our answer here is D. Okay, so now let's talk about models of inheritance, right? We have autosomal dominant, autosomal recessive, X-linked recessive, and X-linked dominant modes of inheritance. So how do we know and how do we understand what autosomal and X-linked means, right? So you know that we have 23 pairs of chromosomes in our cells, right? So one of these pair is sex-linked, right? It determines your sex. It's either XY or XX for women, right? So 22 chromosomes of these genes, 22 chromosomes are somatic, right? Not linked with sex. So these somatic cells are actually autosomal, and X-linked are sex-linked, right? So let's say when we can say if it's dominant or recessive when we look at the pedigree, right? So if you see that each generation, like multiple generation, get the disease, then you automatically say it is dominant. You see that first population, second, third generation all have the disease, and you say it is dominant. You can say, you can see in this example, X-linked dominant, first generation, second, third, all get the disease, right? You say it is dominant. So now, how you say if it's autosomal or X-linked then, right? You just look if it's linked with sex or not. Like here, you can see mother is affected with the disease, and she passes the disease to her son as well as her daughter. Then you look at the father, and father passes the disease to his son and to his daughter. So you say, okay, then it's not linked with sex and it is autosomal. Then you look here where you see that father passes the disease only to his daughters, right? And therefore you say it is 100% X-linked. So why you look only to father? Because look at this table here. You have one allele, X allele from mother, one allele X from father, right? Here you have XY allele father, XX allele mother. So you get next generation, like 2 daughters and 2 sons. You see here that father can pass his X only to his daughters, and to his sons he passes only his Y. So if son gets X, he gets it from his mother, right? So therefore autosomal X-linked dominant diseases pass to daughters only from their fathers. 100% cases, if they will have, like these parents will have daughter, 100% of these daughters will be affected if father having the disease, right? So let's look if mother is affected, right? Let's say mother is affected with the disease. So she's sick because it is autosomal dominant. She passes her X to her daughter and one to her son. And therefore 50% of this 4 children will be affected, no matter of the sex, right? Because she passes one X to her son and the other one to her daughter, right? One to her daughter, the second one to her son. So here you can see that mother passes to son as well as to daughter as well. So X-linked, you see that father can pass only to his daughters, right? You see some correlation with sex, but here you don't see any correlation in autosomal dominant case. Okay, so when you say it is recessive, then when case gives you healthy parents and these healthy parents have sick child and question asks why, and you see in this pedigree there is some linkage with sex, like only males get affected, right? And therefore you say it is X-linked. And how this happens, you can see this on these tables. So you see that if we have the disease, we have, for example, in recessive case, XX, right? And females have 2 X's, and therefore if it X-linked, it won't affect them because they have healthy X, which can compensate this unhealthy X, right? But in the case of males, they have only one X, and if it's affected, they develop the disease, and therefore only males will be diseased if they get this pathologic X, right? This pathology. And in this table you see that when you have male with the disease and he passes his X to his daughter, these daughters are healthy, but they carry the disease. They are carriers of the disease. If you look at mother, you see that one X goes to her son and the other X goes to her daughter, right? One of her pathologic X's goes to her son, and her son have only one X, and therefore he develops the disease. The other of her X's, pathologic X's, goes to her daughter, but she's healthy still. She is carrier. She can pass the disease to her son, but she's healthy. She's not sick, right? Because she have two X's. So one of her healthy X pass also to her daughter and her second son, right? But these children are healthy because no pathology occurs. Both of their X's, of her X's, for example, in this case, right, is normal, and his X from his mom is normal, and therefore he's healthy. So we say in this case that 50% of her sons, of her two possible sons, will get the disease because she gives one of her pathologic X only to one of two. The other son gets healthy X, right? So now let's move to autosomal dominant and recessive tables, right? So we try to make least amount of affected children, affected with the disease children, and therefore in autosomal dominant case, we take only one affected parent, right? Because it's autosomal dominant, and therefore we get 50% of children being affected. So we get 50% of children being affected when one of the parents is hetero, right? Then in autosomal recessive case, again, we try to make least amount of affected children, and therefore, like here you see two homozygous recessive roommate. Here only one. Therefore, we take two hetero and get 25%. Like one of the four children are affected, which means 25% of children is affected with the disease. So this three healthy children, from these three healthy children, we have two affected. So two of these three children are affected. These numbers are important when we solve questions with Hardy-Weinberg equilibrium. When they ask frequency of heterozygous children, carriers, right? Therefore, you have to memorize these numbers in order to quickly solve them. And if you want to watch for an example of this question, you go back and see previous lecture.